Hi, this is Mr. Waite, and today I'm going to go over the AP Biology session of the Saturday study session for biochemistry. And what you should have in front of you is you should have the student study session packet that looks like this. Please go over the first 2 pages of review. They're very helpful before you go over this. And then you should have behind that the 8 multiple choice questions followed by 2 math grid-in questions, followed by some FRQ questions. So please have that out with you, and let's get started on the biochemistry. All right, first thing we're going to look at biochemistry is this idea of anabolism. And don't forget, as we get up here, we're looking at anabolism as building things up. Okay, so we get here, these reactions require energy, yes. So it says the synthesis of a protein produces which of the following products. Now, I know if I'm making something, it's a synthesis, so the first thing I want to keep in mind is this idea of dehydration synthesis, and that's what I have a picture of right here. And in dehydration synthesis, what we see is a monomer, two monomers are connected, or a short polymer, and one will have a hydrogen end, one will have a hydroxide end. When you combine H with OH, you get H2O, and that water comes out of there. Thus the name dehydration. The synthesis is bringing these two together. So let's see if any of these answers fit. Polymer is the end product of ATP and is formed by a byproduct of the reaction. It doesn't say anything about water in that, so I'm going to skip that. A monomer is the end product. Well, we're not really going to end with a monomer. We're going to have to end with a polymer. A polymer is the end product, and water must be added for the reaction to occur. Unfortunately, we're not adding water to this. This one we are taking water away. So water must be added. Doesn't work. The only one left here is D. A polymer is the end product. That's what we have down here. And water is formed as a byproduct. So that's the one that makes the most sense for question number 1. For question number 2, we're going to look at, this is just a review of dehydration synthesis. Don't forget, we just explained. Synthesizing, water is going to come out of it, formed from these 2 ends, and will make a large polymer. If the opposite of that is hydrolysis, we're going to break something using water. We put water into that, and then obviously the hydrogen will go over here, and a hydroxide will go on this end. So 2 opposites that we're looking at there. All right, examples of dehydration synthesis. Common one is ATP. Don't forget, before ATP is ATP, it's in this form of ADP with 2 phosphates. Phosphates have a very negative charge. They don't like each other, so to add a third one onto that would be a tremendous amount of stress. We're able to do that by placing lots of energy inside of there. We get our ATP molecule with 3 negative phosphates. It's very unstable, and when it breaks, it's going to release lots of energy. Another example of dehydration synthesis and hydrolysis. All right, we look here, question 2. Question 2 is concerned about if the water in which of these organisms becomes depleted with nitrogen, what would be the most likely result? So for this one, we have to know which of these compounds right here has nitrogen inside of it. So if we do that, we go back and review carbohydrates. Carbs have carbon, hydrogen, and oxygen. Lipids are mostly carbon, hydrogen, oxygen. Sometimes they'll have the occasional phosphorus. But proteins over here, they are carbon, hydrogen, oxygen. They can also be nitrogen and sulfur. And because they have this nitrogen inside of them, they're the one that's the most effective. Are most affected. So when I look at this, I go back through and look at these. It says the dry mass will contain, this one deals with carbohydrates, which we said is not going to apply. Let me go back to that image that I just had a little bit ago. Carbohydrates, no, they don't have nitrogen. Organic compounds, that's all of these are organic compounds, anything with carbon. So these are all organic compounds. Percentage of the proteins will be less than 25%. Yes, if the nitrogen is going down, then we'd expect the proteins to go down as well. So C is our answer for that one. All right, let's go to the next problem here. C is the correct answer. Question number 3. It says membrane fluidity declines under cold conditions. Muscles move more slowly at decreased temperature. However, when organisms get cold, some organisms can change the composition in order to maintain the movements of the nutrients. In other words, they're going to maintain or increase the fluidity. Okay, fluidity. So we want to find something down here that's going to help increase the fluidity. As we're looking at this, it's going to be helpful to keep this in mind, our picture of the membrane. And what we see is, here is, don't forget, membranes are made of phospholipids with a head and 2 fatty acid tails. We'll also remember that for these, if I get a closer picture of that, I gave the answer away, I didn't mean to do that, sorry. What I was trying to show you was, don't forget, this part is very nonpolar, the tail's nonpolar, the nonpolar fly zone, and this part is polar. So we're going to look at something. When I go back up here, it says, which of the following is most likely structure to decrease temperature? I'm sorry, I kind of jumped ahead on this one. Let's just go back to this. To maintain fluidity, these guys right here, this is going to be our saturated hydrocarbons inside of the tails. Over here we have unsaturated hydrocarbons. And if we're going to allow something to be a little bit flexible, it's going to need to look more like this. So if I go back and look at the answers, replacing saturated fatty acids with unsaturated fatty acids, yeah, these unsaturated fatty acids, this one makes sense. Let's check the other ones. Increasing the amount of saturated fatty acids, no, that's going to make it hard and more rigid. Replacing the shorter fatty acids with longer fatty acids, that's just going to give you a longer, more thick membrane. So we're not going to do that one. And increasing the amount of membrane, carbohydrates, don't forget, can be embedded inside here, but that's not necessarily going to help with our fluidity as well. So let's go to the next one. Just as a review, we looked at that, how the unsaturated hydrocarbon tails inside the fatty acids contribute to the fluidity of the overall membrane. Question number 4, what we're looking at, directionality influences structure and function. This is a very important part of the AP test. Which of the following statements is incorrect? Guys, when you're taking the AP test, make sure anytime it says incorrect, circle it, highlight it. You can draw on the test itself. We are looking for the one that is false, not the one that is correct. So do nucleic acids have a 3' and 5' end? Yes, that's true. That's not going to be our answer. Do proteins have amino end and a carboxyl end and peptide bonds? Yes, that's also true. So that's not our correct answer. The nature of bonding between carbohydrates monomers determines the structure and function of the polysaccharides. Yes, that is true as well. So I am going to say that's not going to be an answer. Over here, lipids contain a hydroxyl end. No, they don't contain that. That's going to be our one that's false, so this is our correct answer right here. All right, if I continue on over here to the next one, question number 4, and it says, that's explaining what we just said. Here's directionality of what we just talked about. Don't forget, DNA has that 5' and 3' end right here. That's an example of directionality. Let's go to the next one, and another thing that we should consider when we're looking at this, influence on proteins. So when we're building up our amino acids through dehydration synthesis, one thing we already talked about earlier is one end will have a hydroxide end, the other one will have the hydrogen. And it's these two ends that have to come together right here, because that's going to form HOH, which is our water. So they have a directionality associated with them as well. Let's look at the function of carbohydrates. Basically what we're looking down here is these two guys over here, the starch and the glycogen, are helical shape, where these over here, the cellulose, has more of a straight shape. So that affects their structure and function. Let's look at the next one. Question number 5, structure and function of polymers are derived from the way their monomers are assembled. Basically it says, which of the following diagrams correctly depicts the monomer of DNA? So let me just move this right here, and we've got this. First of all, you should go through all these and know what most of them are anyways. And what you can see on this one, if I put this right here, I'm sorry, let's get this back to the right spot. This should be obvious right away. This is our 1, 2, 3 phosphates. This must be a molecule of ATP. If I go over here, let's go down. This one right here is a form of cholesterol, and that might be by something that you just memorize. Over here, this one, we've got our rings, and anytime we've got those carbon rings, that indicates some type of a carbohydrate of some kind. In fact, look up here. Here's another carbohydrate embedded in here. This one is actually what we're going to be looking at for this one. Let me change this. Is sucrose. I'm sorry, I had to double check that. But let's put it back on this one and let's look at closely at this other one. Something important to remember, DNA is made of a phosphate group, a sugar group, ribose specifically, or deoxyribose, and then some type of a nitrogen base. This is a purine right here. That's what we need to remember, so that's our correct answer. If we go to the next slide, what we see is that's the right answer. Again, just emphasis that we have a sugar, phosphate, and a nitrogen base. It's good to know what the different nitrogen bases are as well. Purines are larger, pyrimidines are smaller. All right, let's keep going with this, and let's look at the carbohydrate, the monomers, the protein monomers. We're looking at this again to show us this is, we're just reviewing that carbohydrates can have monomers as well. Obviously, we build upon these. The big emphasis point is monomers make polymers. So we can make much longer chains of these guys if needed. Question number 6, which of the following amino acids is the most likely to be found in the region of a transport protein interacting with fatty acid tails of phospholipids? Guys, just good time to review what is a phospholipid down here. They have a polar and they have a non-polar region associated with them. And the tails down here, which it's asking us about, the tails of phospholipids have to be non-polar. Now, this question is a little bit difficult because it requires you to know the R group of each of these amino acids. And guys, this is another good thing. Just recognize here's our amino group, here's our carboxyl group. On each of these, they are the same. The only thing that makes these different is the R group right here. So this question's hard because it would require knowing the R groups. I don't think the AP Biology test is going to be that specific, but I'll throw this in here. And what this is showing us is that up here, leucine is the one that is non-polar. Okay, and if it's going to be in the fatty acid tail of the phospholipid, which is also non-polar, that one would make the most sense. But again, I think that's a pretty detailed question. Let's move on to the next one or see if there's any review that we're supposed to look at. Our polar, non-polar components. Again, I've highlighted it, the amino group, the carboxyl group, and the R groups. I think those are the important things. And question number 7 says up here, the graph on the right depicts effective enzyme on activation energy. Which of the following description best portrays the portion of the curve labeled B? So we're looking specifically at this one right here. It doesn't tell us on this one, but if this one is going faster or it's a shorter reaction rate, a shorter activation energy, this must be the enzyme catalyzed reaction versus the non-enzyme catalyzed reaction. I'm going to throw up a diagram up here just to remind us what these are. Again, this is showing us that this is with an enzyme, the red. This is without an enzyme, the black. And it's specifically asking us the enzyme, the graph, the effect of an enzyme on activation energy. So when we look at this one, letter B right here is the activation energy for this enzyme catalyzed reaction. If I look at my choices, activation energy that is not enzyme catalyzed, that seems like that's not a good choice. The one that is enzyme catalyzed, but let's analyze the rest of these. The total delta G, or the Gibbs free energy of the catalyzed, no, the total free energy is going to be what's given off. So we see down here, this part is labeled as the delta G. So that would be C, total delta G. Nope, that's the same thing down here. So not either of these. The correct answer on this is B. Let's go to the next one really quick. And we looked at that diagram. This is important to talk about. It's this idea that enzymes and substrates have to fit like a lock and key mechanism. I've got just a few more seconds here, so we're going to analyze question 8 quickly. Question 8, what it's telling us is it says product D is able to occupy the active site of enzyme number 2. So if it's able to occupy this, that could prevent this reaction from happening here. And so their answer on this one is that C is acting, is acting as a competitive inhibiting molecule. Guys, just really quickly, if I can, last few seconds, competitive inhibitors This is an example of negative feedback. We'll talk later.